Good morning, everyone. My name is Stéphanie Vargas, and first I would like to thank the organizers for letting me present some of the work that we've been doing with the aims of developing a therapeutical agent for treating neurodegenerative diseases. This work was done in Collège de France and in collaboration with the startup company BrainEver, and is based on the observation that the homeoprotein NGREL1 is a neurotrophic factor for motor neurons. Due to time, I'm only going to present you a very general overview of our work, but please feel free to ask questions after the talk or during the breaks. We will be very happy to try to answer them. So let me start with the basis. In our laboratory, we are interested in a very special family of transcription factors encoded by homeogenes. The proteins encoded by these genes contain a highly conserved DNA binding domain, the homeodomain, that enables these proteins to exert key cell autonomous functions through the modification of transcription, translation, and epigenetics. But homeoproteins can also transfer between cells thanks to the secretion and internalization motifs embedded within the homeodomain. And once internalized, they modify the activities of the host cells at these three levels: transcription, translation, and epigenetics. Of the homeoprotein family, we're interested in the NGREL homeoproteins. Previous work from the laboratory has demonstrated that NGREL1 is an important survival factor for dopaminergic neurons in the substantia nigra pars compacta. So we wondered whether NGREL1 was important for other neuronal subpopulations, such as motor neurons. In the spinal cord, NGREL1 is expressed during development, but it is also expressed in the adult in the B1 subtype of interneurons that include Renshaw cells. As you can see in this RNA scope, where NGREL is in green, calvindin, a marker of Renshaw cells, is in blue, and CAT, a marker of motor neurons, is in green. NGREL1 is only expressed in the B1 interneurons, including Renshaw cells, but not in the motor neurons. So our first important observation was the fact that the NGREL1 protein is present in motor neurons. As you can see in this combination of RNA-scope and immunohistochemistry, the NGREL1 protein here in green is present in the cells that produce it, B1 interneurons, but it is also present in CAT-positive cells, suggesting that motor neurons import the homeoprotein NGREL1. But why would they do that? Why would motor neurons want to have NGREL1? Is it important for their physiology? Is it important for their survival? Our hypothesis is pretty straightforward. If NGREL1 is important for motor neurons, if we take it out, motor neurons should suffer. So to test this hypothesis, we took advantage of the NGREL1 heterozygote animals that lack half of the normal NGREL1 gene dosage. As you can see here, in young adults, but also very old mice, we have half of the normal NGREL1 gene expression. Now, as we all know, and is represented in this scheme, wild-type animals have normal numbers of healthy motor neurons that innervate their muscle targets, allowing these animals to perform in different motor tasks. However, when we analyze our heterozygote animals, we observed that as early as 2 months of age, these animals show motor deficits. They start to be weak, and this weakness progresses as the animals age. We think that this could be due to deficits in the muscle, so we analyzed the neuromuscular junction of these animals, and we observed that at about 3 months of age, there is an important denervation of the neuromuscular junction. This denervation could be just due to axonal detraction or actual motor neuron death. So we analyzed the amount of motor neurons in these animals, and we observed that at about 4.5 months of age, there is an important death of big alpha motor neurons. So these results show that if you take half of the normal NGREL1 gene dosage, you have weakness, neuromuscular denervation, and alpha motor neuron loss in a dying back pathology. And this shows that NGREL1 is indeed a survival factor for spinal motor neurons. But since motor neurons don't produce NGREL1, we propose that this has to be a non-cell autonomous effect. And to test this hypothesis, we used a single-chain antibody strategy, which consists in the injection of a virus that encodes for the single-chain antibody under the GFAP promoter. This means that the single-chain antibody will be expressed by astrocytes, secreted, and it will capture NGREL1 in the extracellular space, preventing it from entering its target cells, including motor neurons. The first thing that we observed is that after the injection of the virus, we have a reduction in the amount of the NGREL1 protein in those animals injected with the single-chain antibody, and not in those animals that were injected with a control virus that express a mutated antibody that doesn't bind NGREL. When we analyze the behavior of these animals, we observe that 8 weeks after the injection, these animals start to get weak. And again, this weakness progresses as the animals age. When we analyze the neuromuscular junction at the end of the experiment, 24 weeks after the injection, we observe that there is an important denervation of their end plates and a considerable amount of neuromuscular motor neuron death. These results show that neutralizing NGRL1 transfer is sufficient to induce motor neuron death in wild-type mice. So NGRL is important for motor neurons. If you take it out or you neutralize it, motor neurons die. Are we able to restore the phenotype? To answer this question, we intrathecally injected human NGRL1. And the first thing that we observed is that 24 hours after the injection of human NGRL1, the exogenous protein is preferentially captured by motor neurons, suggesting that there is a specificity in this system. Now, we established our therapeutical window at 3 months old because remember, mouse are weak, we have denervation, but motor neurons are still there, albeit they might be sick. When we analyze the behavior 1.5 months after the injection, we observed that those animals that were already weak regain their strength, they reinnervate their end plates, and importantly, we avoid motor neuron death, showing that NGRL1 injected intrathecally is preferentially captured by motor neurons and can prevent their degeneration. Moreover, this effect lasts for several months. As you can see here, before the injection, animals are weak. They regain their strength. The effect lasts for about 8 weeks after the injection, when animals start to get weak again. And although they don't perform as good as the wild types, they do much better than those animals that were never injected. This is also the case for the amount of fully occupied end plates and the number of motor neurons at the end of the experiment. Now, from a therapeutical point of view, it's important to note that we can prolong this effect by providing a second injection. As you can see here, those animals that only receive one injection start to get weak again, whilst those animals that received a second injection have a prolonged protective effect. This, again, is also the case for the amount of innervated end plates and the number of motor neurons. So I hope that by now I have convinced you that NGRL1 plays a pivotal role in motor neuron survival, that if you take it out, you have degeneration of motor neurons, but that we prevent this phenotype by providing exogenous NGRL1. And more importantly, that simply by neutralizing NGRL1 in the extracellular space, we can promote motor neuron degeneration. Now, the next question is how? What is NGRL1 doing? And this is a big, ambitious, and very important question that we are starting to try to answer. And as a first approach, we performed a bioinformatical analysis that gave us a list of genes that are targets of NGRL1, but also interact with the mainly associated genes for ALS. Of this list, we were interested in P62 because it has been demonstrated that it's a marker of ALS and because it's a very important protein for autophagy. So we tested the amount of P62 in our models. And our first observation really surprised us because we saw that in the wild-type animals, simply by the fact that they are aging, we see an increase of the amount of P62. This suggests that P62 can be an aging marker. This, remember, it's very important because Parkinson's disease, ALS, and other neurodegenerative diseases have a very important age component. Now, when we observe our heterozygous animals, we see that there is an increase of P62 at 3 months old, suggesting that these cells might be aging faster and have autophagy deficits. The interpretation of 4.5 months old and 9 months old, it's a little bit more complicated because remember, we have already lost 25% of our motor neurons here and almost 50% of our motor neurons here, meaning that what we're measuring is the amount of P62 in those cells that survived. And these cells might be less sensitive to the loss of NGRL1. What is important is that we are able to reproduce these results, again, simply by neutralizing NGRL1 in the extracellular space. There is an increase in the amount of P62, meaning that these cells might also be aging faster and they might have autophagy deficits. Now, we know that there is a lot of work that we need to do in order to elucidate the mechanism of NGRL1. We are currently testing other ALS markers like TDP-43, STAT-MIN, PAPB, but we really are convinced, based on the evidence of our laboratory and other groups, that NGRL1 is a very interesting target for developing therapeutical strategies for motor neuron diseases. And on this high note, I would like to end my presentation, not without thanking everybody that made this project possible, the people of Collège de France, the people of BrainEver, the company that I'm part of, our external collaborators, administration, microscopy, animal facilities, and of course, thank all of you for listening. I'm very happy to take some questions, sorry.
Questions, please?
Thank you. Do you have the same story for central motor neuron?
We have not tested what happens in upper motor neurons yet. The thing is that the heterozygote mice also has death of dopaminergic neurons in the substantia nigra. So we currently have a project where we are analyzing the motor cortex of the heterozygote mice. Right now, we have not looked at them, but we don't think it is necessarily a cell autonomous effect because there's no NGREL1 in there.
Thank you for all those data. I have a couple of questions. The first one is extremely naive. If NGREL1 is so important for motor neuron survival, is there any explanation why those neurons do not express it by themselves? And the second question is regarding the gene targets, and you mentioned this at the end of your talk, but I haven't understood whether these are chip-seq experiments in motor neurons or how you ended up with these genes. Could you further explain? Thank you.
Yeah, so for the first question, I don't think we can find a way to explain why motor neurons do not express it. It might just be an evolutionary thing, and for that we would need to look in other models that are less evolved, probably vertebrates or Drosophila or something like that. I mean, we know that it's not expressed by mammalian motor neurons, but it could be the case that other less evolved systems express the protein. We don't know, we would need to see. And why they don't produce it, it might be, you know, that they can get it from the other cells. So, I mean, if you can get something from other cells, why would you make it? You know, like, you don't use your energy and you just take it from other guys. But honestly, we don't know. It would be great if someone has an idea of how could we prove that they don't produce it, even though it's very important for them. For the bioinformatical analysis, if you want, maybe we can talk together. It's really the results that were taken from the data of the dopaminergic neurons in the substantia nigra. So we get there the genes that are targets of NGREL1, and then all the other stuff was basically just cross-reference with the results of other groups of what genes interact with genes related to ALS. But we can discuss the pipeline and everything if you want.